Module 5, the muscular system, histology and physiology. Skeletal muscle structure. To study skeletal muscle, you need to first learn about its structure. Muscles do not directly attach to bones. Instead, they are attached by tendons. Remember that tendons are made of dense, regular connective tissue, meaning that there are lots of collagen fibers and they're oriented in a parallel-like, ordered pattern. The muscle belly is where the actual muscle cells are. Each of the muscle cells is called a muscle fiber because they are elongated in shape. Each of the muscle cells is covered in a layer of collagen called the endomysium. The muscle cells or muscle fibers are grouped into bundles called fascicles. So a bundle of muscle fibers is just a bundle of muscle cells. The fascicles are surrounded by a collagen layer called the perimysium. Additionally, all the fascicles are held together by yet another collagen layer called the epimysium. Sometimes the epimysium is referred to as the fascia, but that term is a more general term referring to tissue sheets lying beneath the skin. So just remember that the collagen layer that wraps around the fascicles is called the epimysium. I mentioned that the muscle fibers are elongated. They extend until they attach to the bone, but they are attached there by the collagen fibers of the endomysium that is wrapped around them. The fibers continue out past the muscle belly and straighten to form a tightly packed tendon made of dense, regular connective tissue. In fact, the collagen fibers of the tendon extend to the bone and become a part of the bone. That's how tightly attached tendons are to bone. They intertwine with the bone collagen fibers and are really anchored in. This means that every single muscle fiber is firmly attached to its respective bone by tendon that forms a continuous connection with the collagen within the bone. You can see that blood vessels are located among the fascicles of the muscle body. They need to be there in order to provide oxygen and nutrients for the muscle fibers to contract. They also are necessary for removing wastes and carbon dioxide gas as the cells produce them. Muscles also require nerves in order to be active. The nerves stimulate the muscle fibers to create movement. If there were no nerves, or if those nerves were unable to work, the muscles could not function. As I mentioned earlier, a muscle fiber is just another word for the muscle cell. It's just very long and thin. In a previous section, I also told you that skeletal muscle fibers are multinucleate. Their nuclei are positioned right below the sarcolemma. That's what we call the plasma membrane of a muscle fiber. It's given a special name because, as you can see, muscle fibers are very specialized cells, looking and behaving quite differently from many other cells in the body. The prefix sarco actually means flesh or meat, so sarcolemma is a membrane around the flesh. In fact, the sarcoplasmic reticulum of muscle fibers is just the term we use for their endoplasmic reticulum of these special cells. Why do we have to use these special names? Well, you can already see how these cells are unique. The sarcolemma has very small tubules that form pits on the membrane. These are called T-tubules, and they extend down into the sarcoplasmic reticulum. A T-tubule's job is to send the action potential signal from outside the cell down into the sarcoplasmic reticulum of the muscle fiber. I'll talk more about action potentials later, but for now, just know that the T-tubules enable these signals to get into the muscle fibers. Within the sarcoplasmic reticulum are areas filled with myofibrils. These are long, thin, thread-like structures that travel from one end of the muscle fiber to the other, but they are so thin they are only a few micrometers in width. They're actually organelles within the muscle fiber or muscle cell that are responsible for contracting. The myofibrils are organized into groups within the muscle fiber, and that's what gives skeletal muscle tissue its striated appearance. Though the myofibrils are thin, they also can be broken down into two types of filaments, actin myofilaments and myosin myofilaments. I'll talk more about these in the next section. And one more thing about muscle fibers. They contain many mitochondria. Remember that mitochondria are the powerhouses of the cell. Because muscle fibers use up lots of energy, you should expect them to need lots of these organelles in order to provide it. Module 5, the muscular system, histology and physiology. Experiment 5.1, skeletal muscle histology. In this lab, we're going to take a closer look at the skeletal muscle tissue. And we need for that our human striated muscle slide. And let's take a look at that under 40 power. Skeletal muscle is actually pretty easy to identify under the microscope. It stains nicely, and we'll be able to see those individual muscle cells. So at 40 power magnification, go ahead and center your slide, and then let's move it to 100 power magnification. Focus that and center it. And now you should be able to get a good look of the muscle at the muscle fibers. When I say muscle fiber, it's really a muscle cell, a single cell. Each fiber is a single cell. There's dark spots that you can see within the muscle fiber, and those are the cell nuclei. They stained darker than the cell itself. So each strand, which looks like a layer almost, but each strand is a single cell. That's a muscle fiber. Notice how long those things are. And think back to when we took a look at the skin cells and how those were kind of cube-shaped or oval-shaped. Notice how long these are in comparison to those. These are very elongated by design so they can do their job, but they're very different from epithelial cells. At this point, we want to go ahead and increase our magnification to 400 power. And let's take a closer look. That looks great. All right. Now I want you to take a look at the nuclei, now that we can see them more closely. Notice there's more than one nucleus in a muscle fiber. That's because they're multinucleate. They have many nuclei. And notice the position. They're not found smack in the center like a fried egg might look like, where the yolk is in the center. These are located along the edges of the skeletal muscle fibers. And so identify the placement as well as the shape and the number of nuclei when you make your illustration in your notebook. Now, at this point, we want to finally look at there's something unique about these cells. There's dark bands that repeat over and over and over again in these cells, and these are the striations. We're going to talk more about that in an upcoming section on what those striations are, but it's due to a specific arrangement of proteins within a muscle fiber that enable our muscles to do something amazing like that. And so we'll talk about how those things work. But notice the striations in those cells right now and put that in your illustration that you make as well. So make the drawing, identify the magnification, and label those things that we just discussed in your lab notebook. And when you've done that, go ahead and clean up everything and put it away.